Good morning, good afternoon, good evening, everyone, and welcome to today's webinar co-organized by the ITU WHO Focus Group on AI and Health and the Department of Biomedical Informatics at Harvard Medical School. My name is Matthias Graschel. I'm a physician and researcher working with the Focus Group on AI for Health, which is co-hosted by ITU and WHO under the umbrella of AI for Good. AI for Good is the UN's leading initiative around how AI can help achieve the Sustainable Development Goals. If you're interested in what any of these initiatives are doing, please check out the links or find it on Google. And we're always looking for people to join the effort in case you would like to join. In this series on AI and health, we try to cover relevant aspects and challenges in research around AI. For example, last time we heard Siad Obermeyer from UC Berkeley talk about bias, and that's something we'll surely hear about today as well. And we'll have a couple of webinars in the next months from Global South scholars from South America, for example, to give their perspectives. But today I have the privilege of introducing two amazing speakers who are going to talk about the disorderly world of diagnostic and prognostic models for COVID-19. Laure Wynants is an assistant professor of epidemiology at Maastricht University in the Netherlands, and she had studied biostatistics and graduated with a PhD from the Catholic University of Leuven in Belgium. Since March 2020, she leads an international consortium to systematically review models for COVID-19, which has been awarded by several prizes. And the resulting publication, A Living Systematic Review, has, and I just checked today, now 1,522 citations, which is a lot in a year, and which showcases how important and impactful this work has been. We also have Maarten van Smeden today. He's a statistician working at the University Medical Center in Utrecht, also in the Netherlands. And his work is focused on the development and evaluation of statistical models and methods in general and how to use them in clinical prediction. If you're looking for a healthy dose of science sarcasm every day, I recommend you follow him on Twitter. Dear audience, please ask away during today's talk. Please submit your short questions in the Q&A tab. The shorter, the better. That'll make it easier to moderate the questions and make sure we can ask Laure and Maarten as many questions as possible. All right, with no further ado, Maarten, you're going to go first. Please take it away. Thanks for being here.
Thank you very much for the kind invitation. So it's a privilege to talk about something that has been in our lives for 1.5 years, COVID-19. And especially about the things we have been doing, Laure and I, and a large team actually, looking at diagnostic and prognostic models for COVID-19. Next slide, please. So what I'm going to talk about is, first of all, what do we mean with AI type of models that we are talking about? Why we should review them. I will talk a little bit about the how and the when, the methods that we used to do our review, something about the results, and then Laure will take over to talk about what is bias and some discussions on the lessons learned through this project. Next slide, please. So what type of AI models are we talking about? Next slide. Well, there's a lot of discussion about, or you can have a lot of discussion about how to define AI. For example, here from the GRC policy report. In a broad sense, we could say that AI can be understood as the study of how to produce machines that have some of the qualities of the human mind, such as the ability to understand language, recognize images, solve problems, take decisions, and learn. So a very broad term, obviously, AI. Next slide, please. And what we are actually looking at is AI to be used for diagnostic and prognostic models. And the idea of those models is that they are used for predictions on individualized patients, on whether they have a certain disease, so probability of having a certain disease, typically, or experiencing some health problem over time, let's let's say, for example, myocardial infarction in 10 years or much shorter. If you are admitted to the hospital with COVID-19, will you survive? And of course, that's based on many inputs, depending on the model, which can be just simple demographics up until very complex images or diagnostic tests. And it's very data driven. And a good thing to realize here is that this is not new. We have been developing these models since at least the 1950s. So it's actually something we do that is very common. Next slide, please. But what has changed is, of course, the upcoming of artificial intelligence and machine learning in the field of medical prediction, in the field where we are interested often in making diagnosis and prognosis. It's interesting to see that one of the major things that have changed is probably the terminology. What we call prediction. In machine learning, you often hear the term supervised learning. Target is our dependent variable, ground truth, that we used to call gold standards. Features are what we call predictors. Learning, we used to call fitting. Weights are parameters. And very important, if we talk about development and validation of AI algorithms or prediction models in general, machine learners often talk about training and testing. And simple, even simple parameters like sensitivity and positive predictive value, which are very common to the medical audience, are something called recall and precision. Next slide, please. So we're talking about artificial intelligence in a field that is much, much older than artificial intelligence, or at least since a new interest in artificial intelligence. So the question is, why look at these models anyway? Next slide, please. Well, that's one of the reasons is just because they're everywhere. This is a small grab of some recent papers, or at least they were recent in 2019. These were just a few papers that were published within a month, talking about leveraging machine learning techniques, even multi-center machine learning studies, roadmaps, infrastructures. A lot of literature these days in the medical journals is about AI and machine learning. Next slide, please. And there's a lot of promises also. For example, at least in the popular media, that doctors will be replaced by a hospital AI system, offering better diagnosis and less arrogance, which sounds great. Next slide, please. And of course, it's very nice to think about what AI can do in medicine if you compare it to the tech company business models, the very successful tech company business models like Netflix, Google, and Amazon. Next slide. And actually, we do. We often compare ourselves. These are just a few examples of medical literature, or at least scientific literature, where we try to compare ourselves to Netflix, Google, and Amazon. Next slide, please. And then, of course, came COVID. Everybody knows about COVID, so I don't have to introduce it. What was interesting was that actually from the start, we saw a lot of news, and this is March. These are all this news from March 2020. So right at the start, at least for here in Europe, this was the start of the pandemic, where there were already reports that there were hospitals employing systems screening 1,300 patients per day using AI. Or even in Italy, of course, where the real problem started. Apparently, some interventions AI was used to battle COVID, and also tools to help in the UK, and news about Europe. And I think this is not just theoretical. There was a lot of also discussion about who to protect. So the vulnerable people for experiencing a severe form of COVID. they had to be shielded in some way, and there was a lot of use of AI also for this. Indeed, there were also algorithms developed to predict COVID-19 complications in emergency use who actually got FDA approval, and also artificial intelligence to improve outcomes of critically ill patients. All got FDA approval, and it's pretty new because we haven't seen those approvals for this kind of algorithms in the past. Next slide, please. So why would you develop these models anywhere? Well, the idea obviously is to develop good models, good AI-based prediction models to improve care and reduce costs, getting diagnosis quicker, more efficiently, and with less error, for example, or being able to predict future health in individuals. And one of the things, of course, you can do with that kind of information is to allocate financial or healthcare resources when there are scarce. Next slide, please. Unfortunately, there's also a downside of using these models. One of the things is that there's at least a worry that inaccurate predictions can lead to harmful decisions, and I will have an example very shortly. And actually, we knew about an earlier pandemic, the H1N1 pandemic, where ICU scores, which were simply predictions, performed very poorly in this new pandemic with individual patients. Next slide, please. So this is that example where you can, which really nicely shows that if you have a model that makes wrong predictions, you can be worse off than having no model at all. So an interesting paper. It was actually a trial looking at deep learning, so AI-based assistancy for histopathology classifications of liver cancer. And what they showed was that actually with that deep learning assistant, the odds of making a correct diagnosis improved fourfold. So that's quite a big improvement. But that was conditional on the fact that the model was actually making the right predictions. And what they also did was actually switch the predictions to the wrong predictions. And what you then see is that the odds of a pathologist making a correct diagnosis actually decreased quite substantially, only 1/4 of the odds. So this clearly shows that if you have a model that makes incorrect predictions, you run the risk of making worse classifications than with no model at all. And of course, this might have influence on your patient's health. Next slide. So in the end, of course, as any model or any therapy, it's all about risks and benefits of these models. Next slide, please. So I want to talk about the actual review that we did. We'll talk about the methods a bit. Next slide. So this is the paper that was already mentioned in the introduction. It's a paper published in the British Medical Journal called Prediction Models for Diagnosis and Prognosis Review of COVID-19, Systematic Review and Critical Appraisal. It's the first BMJ living review ever. So we're very happy with that. And actually, we started with this project in March 2020, so right at the start. And already in April, we got our first publication. So it was a very speedy process of getting the first review published. And at that time, we didn't think about living reviews, but after that was published, we were invited to make it a living review, meaning that we are updating this review on a regular basis. In fact, we updated it once in June, once in July, and the last time was last February. Next slide, please. So the types of articles that we looked at was peer-reviewed articles through PubMed and Embase, and we looked at preprints, so papers that were not yet peer-reviewed, only until update 2. That had to do with the amount of literature that we actually had to go through. Next slide, please. And we distinguish between 3 types of models. The first types of prediction models are diagnostic, meaning they look at whether a patient X is infected or whether a patient X has COVID pneumonia. Or prognostic, so for example, patient X will die from COVID-19 or will need respiratory support. And the third category is currently healthy person X will get a severe COVID-19, which we will call general population models. Next slide. So how did we do this? Each article that we found was screened by 2 independent researchers. If more than one model was described in an article, every model was extracted separately, so reviewed separately. And to help us, we used CHARMS, which is more or less a form that helps you extract data from this type of models, diagnostic and prognostic models, and Probus, which is a risk of bias tool, also specifically for diagnostic and prognostic models developed. These were developed before we started our review, so we could use it. Next slide, please. So the results. So the first part is, of course, about the number of records we had to screen. We had to go through 37,400 articles. We actually used a little bit of AI to help us with that. Then the next step was we had to go through 445 papers. Some of those were also excluded, and the reasons are seen at the right of the figure. And in the end, we ended up with 170 studies and 236 models, either diagnostic models, prognostic models, or for the general population. So actually, we only got 11 models for subjects at risk in the general population, 118 diagnostic models, and 107 prognostic models. Next slide. So some of the, to give you an idea of where the data came from, well, most of the data that we used to develop the models came from a single country, most frequently China, some Italy, some US, some South Korea. And there were also some international combined data sets, and there were also a few with an unknown origin of data. Next slide. We saw a lot of models that were developed on hospitalized patients, much less in patients, for example, with the general practitioner, and also a lot of models that we didn't know what the target setting was, or what the at least the setting was that was targeted when developing the prediction model. And it also has to do with the targeted population, and many of them based on confirmed COVID-19 patients or suspected COVID-19 patients. Of course, a lot of these models were actually developed before testing was widely available. And also some unclear. And I think a very interesting finding is that only 48% of the models we found, 236 models in total, 48%, so approximately half, were available in a format that can actually be used. So you could also turn it around. More than 50% of the models that were developed were not actually available to be used at all. Next slide. Now, this is an interesting figure. I think not because I like pie charts, because I don't, but in this case, it shows it quite nicely, is that we found approximately 1/3 of the models were based on simple logistic regression. About also 1/3 were based on the more AI types of models, the deep learning algorithms. And then there was still 1/3 of other models like Cox proportional hazards models, support vector machines, random forest, that kind of models. So it's nicely in some way distributed. Next slide. Something about the included predictors that we often found. So what did these models actually use to make the predictions? In the diagnostic models, it was often based on vital signs like fever, flu signs and symptoms, the age of the patient, and with prognostic models also based on age, comorbidities, and for example, vital signs were used a lot. Next slide. We found, of course, a lot of models, and one of the ways to quantify part of the performance of those models is the so-called Cstatistic, also known as the area under the curve or the concordance statistic. And what we found is that they are well, broadly distributed, so to say. So a model that doesn't cannot discriminate, so has a very poor performance, has a C-statistic of 0.5, and the most perfect prediction would be 1. So all these values should be between 0.5 and 1. And what you see is that there's quite a lot of spread. But what you can see in almost all of these examples, or in all of these types of models, that there are models that are nearly perfect, an area under the curve or a C-statistic of 0.99, or even slightly higher than that. Next slide. And this is where I'm going to ask Laure to take over the rest of this presentation.
Yes, Laure, go ahead, take over the mic.
Thank you. Can you hear me?
Yes, we can hear you fine.
So what Maarten showed you is, yeah, of course, for each of these types of prediction problems, we supposedly now have models that work nearly perfectly. But the real question, of course, is can we trust this reported predictive performance? Because if we can, that would solve a lot of our problems. We wouldn't need PCR tests anymore. We would know based on symptoms and with an AI who has COVID and who does not. Whenever, again, ICU beds are too limited for the amount of patients coming in, we would have models to help us that predict very well, that would help us in triage. So can we trust this predictive performance? Well, remember, Maarten told you we did a standardized risk of bias assessment for all of these models. And in this assessment, we actually found that only 4 of the models we looked at were at low risk of bias. 6 were at unclear, and 226 had a high risk of bias. Now that means that there were actually flaws in the design or in the analysis of the study that suggests to us that the estimated predictive performance is probably too optimistic. And you can see that these issues occurred for all the types of models that we looked at. So before we elaborate on this, I of course have to explain to you what we mean with bias. What is bias? And the answer that you get depends on who you ask. Maybe some of you in the audience are policymakers or ethicists, and then your first reflex will be to think about bias in terms of bias and fairness. So we know that AI may sometimes automate or propagate existing biases, such as racial, gender, or social biases. Now that is very important, but that is not what we looked at. If some of you in the audience are acquainted with machine learning, you may think about the bias term in a regression equation, or the prediction bias, which quantifies on average what the difference is between the average of the predictions and the average of the labels, so the true outcomes. Now, this is a very limited way of looking at bias, and that's also not what we mean. Go a little bit further than that and look at what a statistician defines as bias. You will find that they will look at the expected value of the difference between your estimator, so theta hat, and the parameter that you want to estimate, theta. So it's in essence about how wrong you are on average. And note that you can be correct on average, but in individual cases be very wrong. So that's the bias variance trade-off, also very important, but not necessarily what we are looking at. An epidemiologist has a little bit slightly different way of looking at bias, because when they speak about a bias, they speak about the error that led you to being wrong on average. So an epidemiologist says a bias is an error in conception or the design of a study, or in the collection, analysis, interpretation, reporting, publication, or review or the data that leads to results or conclusions that are systematically, as opposed to randomly, different from the truth. And now we're getting closer to what we mean. Bias is very popular among epidemiologists. We actually have a whole catalog of biases, but we want to know the type of biases that are relevant for risk prediction modeling. And in risk prediction modeling, we refer to a risk of bias to occur when there are shortcomings in the study design, conduct, or analysis that could lead to systematically distorted estimates of a model's performance, and usually a too optimistic estimate of model performance. So we think we're predicting very well, but in fact, we have reason to believe that we may be doing worse. And to detect such problems, such errors or shortcomings, we have the Probus tool to help us. So this tool is a standardized risk of bias tool, which asks signaling questions in 4 domains. So 4 domains in which we could go wrong. The first one is the participant domain. So who actually contributed data to the model? The second one is a predictor domain. So what are the inputs that go into the model? How are they measured? When were they measured? How were they defined? The third domain is the outcome. So what are we actually trying to predict? And is this a relevant and well-measured outcome, well-chosen outcome? And the last one is the analysis domain. So any analysis that we do to build our model or to test it, what are the things that can go wrong there? So among these 4 domains, you see that there are actually errors or problems with all domains. So for example, the participant domain, the patients on which data was collected, or the people on which data were collected, they are often not representative of public health or clinical targeted patient populations. So not representative of the people they are intended to be used on. In terms of the predictors and the outcomes, also these are sometimes inappropriately chosen or inappropriately measured. An example that I can give for the outcome is that some models pretended to predict COVID-19 outcomes, but in fact, the data sets did not contain a single COVID-19 patient. This was very early in the pandemic, and they used other data on other respiratory illnesses, but then marketed the model as a COVID-19 model. But really, the most problematic domain was the analysis domain. So the top 3 errors are in this domain. So there are frequent problems with the sample size, with the model performance measures, and with the optimism in the models. So this can be very abstract. So I'll walk you through some of the examples that we found of these errors. So please consider this study. Here, the goal was to build a model to predict mortality. And you see the reference here. It's actually a published and peer-reviewed model. But if you look at the information given in the abstract of the study, you see that the model had data on as few as 19 patients that died to learn from. So that's really not a lot of events for a data-driven algorithm. In fact, if you look at our whole review, you see that the median number of patients or participants included in a study was just 344, so very low. But what really drives your sample size, or what's most important when you're predicting, is the number of events you have. So an event, that's the outcome class that you're trying to predict. So that could be the number of people with COVID-19 for a diagnostic model, or for example, the number of people that died for a prognostic model. And you see that's even lower, 70. So this number of events should always be balanced with the chosen modeling strategy. And overall, 70% of the models that we saw were built with insufficient data for the chosen modeling strategy. So sample size really was an issue. Now let's have a look at model performance measures. What you see here are some plots that demonstrate the performance of a model to predict which patients will need mechanical ventilation. And the right-hand plot is a ROC curve, and you see the gray line, this model has really excellent or very good performance. So a curve that comes close to the upper left corner of the ROC plot is very good at discriminating between patients who need mechanical ventilation and patients who do not. So that looks very promising, but then if you look at the right-hand plot, you see that that's not the whole story. At the red arrow, you can see that the predicted mortality risk of roughly 2% corresponds to an actual mortality risk of 10%. So the model is very badly calibrated. Sorry, not a mortality risk, mechanical ventilation risk. So the model is very badly calibrated. Now, what is problematic is that most studies don't look at calibration. You never see the right-hand plot. You only see the left-hand plot that indicates good performance. So in fact, only 68% of models were appropriately validated in terms of both discrimination and calibration. Now, once you have your predictive performance estimates and you choose them well, there's another thing that goes wrong. Because to avoid optimism in the estimated predictive performance, you need a reliable validation strategy. And what you see here is that the majority of newly developed models were only internally validated, for example, using a bootstrap or cross-validation, or not validated at all. Now, an internal validation can give you reliable performance results, but then you have to be very careful not to leak any information to the test set in your bootstrap or cross-validation procedure. And what you see here is an example of a published model where such leaking took place. So what you see is that these researchers performed the variable selection or the feature selection for their model on the entire database. And only after they chose which features or variables went into the model, they set apart their validation set. So by doing that, you are biasing your predictive performance because you already know what's going to be important in the test set. So not properly accounting for optimism was actually an issue in 53% of models. There are some other interesting problems that were not in the top 3, but I would still like to share them with you because they are noteworthy. So many researchers try to predict in-hospital mortality using electronic health records data. And very often, they don't put restrictions on when the predictors were measured. So in this example shown here, the researchers used the lowest oxygen saturating measured during the entire patient's stay as a predictor of mortality. Now that can lead to very interesting patterns in the data. You see here that a number of patients had an oxygen saturation of exactly 0%. Now I talked to some people and did some research. That's not even plausible in deceased patients. So it's very likely that the oxygen saturation fell below the detection limit of the oximeter in these patients. So the predictor measurements may actually have been made while the patient was already dying or even post-mortem. So that makes the prediction of mortality misleadingly easy. Another interesting problem was the Frankenstein datasets. So in this example, the authors combined lung images from several online repositories, and this means that many imaging studies use images from cases without COVID-19 that came from different sources than the images from the cases with COVID-19. And there are several problems associated with that approach. The first one, obviously, is that the data is not representative of the clinical target population. So in no way is such a set of images comparable to a consecutive series of patients that got imaging due to the suspicion of COVID-19. Another problem is that in such data sets, the same image can be included more than once. And when you then do data splitting to train and test your algorithm, the same image can be included in the training data and the testing data without you realizing it. And that, of course, gives you a biased estimate of performance. And then there are also problems with the cases and the controls differing in many ways other than COVID-19 status alone. So one of the most frequently used data sets on controls contains images from children. So instead of learning to distinguishing between COVID and non-COVID, the model learns to distinguish between kids and adults. So the last problem I want to bring to your attention is that it's often not clear that models, when models are intended to be used, or even in which patients they are intended to be used. So overall, there's very poor reporting. But luckily, I don't only want to bring bad news. I also want to bring some good news. There were 4 low risk of bias models, and these were the Q-COVID models. So these models predict death or hospitalization in the general population. So people who are currently healthy. They were validated in an extraordinary effort in a database of 80% of English adults, and they worked very well. They have been used in policy, so it's one of the examples that Maarten showed in the beginning of the presentation, and they have been modified in the meantime to be used in vaccinated patients. So what should we now, this was a lot of information, I'm sure. So what should we remember from this, or what should we learn from this? Well, first point is maybe you're asking yourself whether Maarten and I are exaggerating. Have we been too critical? Unfortunately, there have been external validation studies in the meantime that confirmed our fears. So they confirmed that the performance estimates were too optimistic. And this plot is from one such study. The colored lines, the colored curves falling from below the horizontal line, show that if you test these models independently, the multivariable models to predict deterioration are actually less useful than oxygen saturation alone. And multivariable models for mortality are actually less useful than age alone. So they really don't predict as well as you might think based on the development studies. So overall, our results do indicate that most COVID-19 models were not ready to be used in clinical practice. And this has led the WHO to recommend not to use any of these models in the clinic. I do have to note that there are other models available that have never been published. For example, in-house developed algorithms in hospitals or proprietary algorithms from AI companies. If there was no publication, no scientific publication, it fell outside of the scope of our review. But we do have some indications that their quality is not necessarily better than what we have seen. So in terms of lessons learned, I have three key lessons. And the first one is that the popularity of prediction modeling has led to a tsunami of models, really hundreds of models for exactly the same disease. And no one can keep the overview of all this. And the second lesson is that most of these models are of dubious quality. So when you want to find a good model, you're actually looking for a needle in a haystack. And the third lesson is that I think, at least, that too much competition really threatens the safety of the patients that you may use these models in. So the obvious question then becomes, how can we do better? Well, I think that our analysis of bias has shown that the first solution is to adhere to methodological standards to prevent overfitting and prevent optimism. The second step is to stop using these very small databases. I think we need collaborations to collect large and high quality data sets. And that these data sets should not just be large, but they should also be representative of the clinical target populations. So that means that you also need to understand the context in which the data was generated. And the third, I think, is that we need to make the code available or the equations underlying the models publicly available. So that makes it possible that an independent party can test models and make head-to-head comparisons of models. And the fourth is that I think we need to work in multidisciplinary teams so that we have models that match a real clinical need and simultaneously are methodologically sound. So in terms of ongoing work, I can tell you that we are still working on the fourth update. Due to the large amount of models that have appeared, we have restricted our focus to only prognostic models, but still we have 251 newer studies. Together, they proposed over 300 new models and also around 200 external validations of existing models. We are also working on validation, multi-country validation, so testing some of the models that have been proposed in international databases. Also, we are validating the models in data from low and middle income countries in collaboration with the WHO. Further on, we have some modeling work or model development work in two separate studies for two separate relevant subpopulations. So the first one is the COPE study, which focuses on prognosis for the elderly. And the second one is a pro-COVID study, which focuses on prognosis in primary care. As Maarten has showed you, most studies have focused on hospitalized patients because that's where the data was. We also need information on patients in primary care. And the last point I can share with you is that Maarten and I also were collaborating on a field standard for artificial intelligence in health in the Netherlands, initiated by the Dutch government. So these are my acknowledgements. A lot of people have made enormous contributions to make this review possible. And in the end, I would like to invite you, if you want to know more, to visit our website. So on our website, we have details on each reviewed model. We also have a layman's summary of our findings, which has been written together with survivors of severe COVID-19. And if you have any questions, we're very happy to answer them now, but you can also contact us afterwards via email or via Twitter. So thanks for your attention.
Thank you so much, Laure and Maarten. Maarten, may I invite you to start your camera again? And I'd like to commend you first on this really important work, on not just doing this work, on also making it accessible on your website to anyone interested. And I think, and also I want to relay one comment by Adrian Wall, who want to also say that it's really useful to get this overview on different biases and how different disciplines look at it. This should be part of every talk, I think, or should be repeated as often as possible. We recommend it. So I think I want to start off the discussion by asking, so when you have data, you build a model, I think there's always been a huge shift sort of between you build your model and you publish it as a scientist and what it takes to really put it into practice. And it seems, based on your findings, it may be even worse with COVID models. Do you think this is a result of looking in March 2020, 3 months into the pandemic, and you had 236 models? Do you think people were too quick, too fast? They jumped on the COVID train? What is your thought on this, Maarten?
Yeah, I think that's part of the problem. Yes, so the early models were, of course, developed very soon and with very small data sets also. So there was definitely a COVID train going on. On the other hand, we also know from earlier literature reviews in other areas that reporting is an issue. And I think one of the things we shouldn't underestimate is that it's pretty easy to develop a model very quickly and to publish those results. And of course, we're still rewarded for our publications as academics. So it's probably there are multiple reasons. Probably, I think the COVID train is a big one. Yes.
Okay. Yeah. So everybody, please submit your questions. I'll kick off with a question about how data privacy laws in the European Union may impede data-driven research. What is, Laure, do you have any thoughts on how that might impede research into COVID using data and models?
Yes, from my experience, it has hindered. It has hindered research. It's very difficult to get your hands on individual patient data or to make the necessary coupling between databases from different resources. That slowed things down. So that was one element. Yeah, I don't know if Maarten has anything to add to that. I can elaborate.
Well, we have seen also in our own situation where we try to validate it in multiple countries, so validate models in multiple countries, and we have experienced ourselves also how difficult it is to actually get together those data sets. And in the end, it wasn't even possible. So you have to really make a lot of effort to do that validation with multiple steps in between. And partly that might be due to regulations, and also partly because people really don't want to share their data. I mean, I think it's two things also.
What would be your ideal scenario in terms of, even if as an individual or as a patient you would like to donate your data for research, how would the ideal scenario look like in your mind? And do you see any investments now after, or like still in the middle of the pandemic, but sort of that regulators or governments see the value of well-collected data sets?
Yeah, I would like to respond to that. Well, we always have to be careful for volunteer biases, the way you put it now. If people contribute their data, the people who are willing to contribute their data are not necessarily representative of the target population. So that might bring in some issues of its own. Have we seen the realization from governments that these data are in silos and are difficult to access. Definitely so. I think there's a lot of communication about European health data space, about gathering data in data lakes. I think it's important to have these discussions, but I'm also a bit careful in having too high expectations of what these data lakes can teach us. So I think this is actually what I was alluding to in one of my last slides. It's not just enough to have large databases, to bring a lot of data together. Really understanding the clinical context in which this data was generated, for me, is really crucial. You have to understand what's in the data, how was it collected, what were the definitions, are there selection biases that we have to be aware of? Because all this is necessary for prediction research to know whether the data is really representative of the setting a model will be used in the end. And if it's not, there's only so much you can learn from those data.
Yeah. Adding on to that, Andrew just sort of made the comment that also the incentive for academics to publish is really wrong. As many papers as you can, and that's on your next grant or tenure panel. That's what probably your assessors will look at. Maarten, do you have any thoughts how to move away from this publishing model? And Andrew is suggesting to have a system in place like ongoing trials.
I think it's very difficult. I mean, it's so part of our science, of course. This and I'm very aware of initiatives that try to improve the situation in science. Actually, Laure and I and some other people just published a paper in the Journal of Clinical Epidemiology talking about exactly this. But it's hard. It's a hard issue, and I have a lot of understanding for people that want to publish their paper and get promoted and get job security and that kind of stuff. And also, I think we shouldn't underestimate that a lot of people have tried to develop a model with the best intentions. So it's not only poor intentions, only career intentions, if they are, if you can even say that those are poor reasons. But also people that really want to but don't have the knowledge, and so I think it's such a big combination of both. But I will not say that I know the answer to solving the biggest crisis, I would say, in science. That's way above my pay grade.
Yeah, publication and industry, all that, of course. Laure, Thomas is asking, do you see a positive trend in the quality of publications now in the 4th edition?
We definitely do see a little bit more low risk of bias, but only a little bit, and a little bit more of unclear risk of bias. But also just the sheer number has gone up, and we excluded category of models that were higher risk than average. So it's hard to say. We did see a lot of interaction with our review. So what we noticed is that people started carefully reporting the things that they realized we were looking at. But sometimes that also uncovered some issues. So they said they did a cross-validation or a bootstrap, but then between the lines, you could read that they implemented it in the wrong way. So they were filling in the check marks. But making mistakes along the way, and it really takes a trained eye to catch those issues, I think.
Imagine. I'd like to zoom back into the science again and ask, maybe first Maarten and then Laure, when predicting a prognosis such as disease course, what is it that you're actually predicting, and should you be doing it at all?
I think it's an excellent question. So there were a lot of different prognostic outcomes. So some of the models were predicting whether you would be admitted to ICU if you just entered the hospital. Other prognostic models were more about mortality, death. Of course, that's also easy to collect data on. And I think that the second part of the question is, should you use it at all? And I think that's a good question, because you always have to question yourself, what if I can predict, what do I do with that information? And one of the challenges, of course, is that we have very limited ability to treat a patient when he or she comes into the hospital with COVID. Of course, there are discussions about allocating healthcare resources, but that's sort of the worst case scenario, of course, where you are admitting or sending patients home with the worst prognosis, maybe. That would be worse, I think. So I hope the models are not used for that. Laure, anything to add, maybe?
Yes. We do know that at least in some cases, these models were used to inform triaging protocols when such, when there would be too much demand for ICU beds. So we know that people in the field have been thinking about implementing these and have developed protocols that implemented these. It's hard to know to what extent they were actually used. And that makes it worrying, because the interpretation of a risk prediction of a model built during the first wave of the pandemic is actually only valid during the first wave of the pandemic. So as soon as care starts to change, or patient management starts to change, or more beds become available, or treatments improve, these risks aren't valid anymore. And it's really hard to know which direction this goes into. And then it really becomes speculation. So is it still useful? Does it do more good than harm if you build your model the correct way? And we, unfortunately, I don't have an answer. I don't know if Maarten has, but I think no one knows. And we need more research to find that out.
Yes. Thanks for this answer, both of you. Yeah. Stefano has a question for Laure, and he's asking about the slides where you talked about how to properly present the performance of your model, not just an AUC plot, but also the calibration curve. If you want to take a minute.
Yeah, yeah, that's a little bit of a technical question. So basically what we want to know is not just do people who die get a higher risk estimate on average than the people that don't die, but also how accurate are these risk estimates. So ideally, so the first thing that you want is that when you apply your model to a complete data set, or all patients within a hospital within a year, if you say 10% will die, then it's actually also 10%, not 20%, not 5%. So you have to compare your predicted risks to what's actually observed. And you can do, as I just said, for a whole database, that's calibration in the large. But to be useful for individual patients, you actually need to look at the calibration for every predicted risk. So for a predicted risk of 5%, do patients with a predicted risk of 5% actually also die at a rate of 5 in 100? And that's a whole lot trickier. So what you often see is that high risks are overestimated and low risks are underestimated, or that all of them are overestimated, or all of them are underestimated to a certain degree. And that, of course, makes it very hard for a clinician to, or for anyone, for a patient, to use and interpret these risk predictions in clinical practice. So this evaluation of calibration, which is ideally done with a calibration plot, the type that I just showed. Well, a colleague of ours has called this the Achilles heel of predictive analytics, and I think that is right. That is true.
As a last question, I'd like to ask, as you mentioned, you're working towards standards in the Netherlands. And here also a plug for the ITU WHO focus group on AI for health, who are creating standards and also an independent assessment platform where you can upload models, have them run and be validated to have sort of an independent assessment. But in your work towards a standard, and this is also a question of Thomas, one of our attendees, how do you go about it? What is your expectation of this effort? And where do you see like these standards in the coming years now that we're still in the pandemic and hopefully after the pandemic?
Shall I start?
Yeah, Maarten, you can start, and then Laure gets the last word.
So I think standards will help, absolutely. And I think one of the issues I think with these prediction models is also related to the earlier question: what are you going to do with it? A lot of them will remain in the publication phase, so it's published and it will never be used. To actually get a model, and I'm sure a lot of attendees know about this, but to get a model into practice, into the clinician's office, so to say, many steps have to be made. So that's not only the expertise that we have about how to develop such a model, how to validate such a model, but also how to develop the right software, how to maintain the algorithms, that kind of stuff. And I think there is a need for standards that describe this whole process. So we have some idea about how to develop those models. We don't adhere to them, but we have some ideas. But especially for the later stages, there's still a lot of uncertainty how to do it. And there's also regulation. So there's EU regulation, there's FDA regulation about these models. And there's also, I think, there's a lot of uncertainty about what regulations the models have to adhere to. So also there's need for guidance and a need for help. On top of the much of the methodological work that has been done since the 1950s already for how to develop those models, because there's a very rich literature, but there's some new things going on too that really need, I think, development of standards. I will stop here and let Laure take over.
Yes, well, of course, I agree with what Maarten has said. What I also observe is a lot of standards being developed now for exactly the same problem. And sometimes that also, well, in some of these standards, certain groups, like the industry, are better represented than in other working groups, or overrepresented, I should say. So, but at least I think it's good to have this discussion, to have all stakeholders around the table and to find answers to these outstanding questions together. So where do we want to go with this new technology, and how can we assure that it's actually that it's actually improving our healthcare or other societal domains, rather than just making it more complicating or more technologically fancy. So yeah, I look forward to seeing where this is going, just like you all, I think.
Sort of, we have 10 standards. Let's make a new one in this way. Okay. So we're at the hour. I'd like to thank Laure and Maarten again for speaking to us today on this really important and really well-conducted study, which is proof by the many citations and attention that you've gotten for this work. And I think it's totally justified. Thank you so much. You can follow Maarten and Laure on Twitter. You can contact them by email or reach out to them or rewatch this webinar today on YouTube. We'd like to start a really quick poll if you would recommend these AI and health series further, if you like this AI and good program. So please hit whatever you feel between 1 and 10 as the poll comes up. And with that, we have reached the end of today's session. Thanks so much, everyone, again, the AI for Good team, and especially Laure and Maarten today. Have a great rest of the day or rest of the morning, wherever you are, and hope to see you next time. Bye.
Thank you.
Thank you. Thanks for the nice questions too.
Selector, rewind selector, rewind selector, rewind selector, rewind selector, rewind selector. Selector, rewind selector, rewind selector, rewind selector, rewind selector, rewind selector.